Hello, everybody. We are on chapter 6 now, and essentially we're diving into the blood part of the cardiovascular system. Some of this is review for you, which is not a bad thing, so let's just hit it. Functions of blood. All right, we already talked about how blood can regulate our body temperature. We also know that the blood is transporting oxygen and nutrients and picking up waste and carbon dioxide. It's also distributing hormones to the appropriate organs where there are target receptors for those hormones. We also know that in our blood we have white blood cells. And those white blood cells, there are some that are on demand, just hanging out all the time, like natural killer cells and macrophages. And there are other white blood cells that are going to be launched in the immune system to help fight infection. Another property that blood has is blood clotting proteins. So there's a series of events that occurs whenever you cut yourself. Platelets are going to rush to the area, some chemical signals are going to be sounded, and then we're going to start a cascade of events that essentially leads to blood clotting, or what you might call a scab. Other ways that the blood is really helping out the cardiovascular system is in homeostasis. So things like pH balance, buffers, the regulation of salts in our blood. Those are all very, very important functions of the blood that is going to help maintain homeostasis in the cardiovascular system. Now, when we think about blood, most of the blood, or just over 50% of the blood, is made up of plasma. So this is essentially this liquid part of the blood. I don't know if you've ever done a whole blood donation, but essentially when you do that, they are going to take out your red blood cells and whatever white blood cells are there, and they're going to put the plasma back. So this plasma is the liquid connective tissue, and it is containing in it what we call the formed elements, which are literally the cells, red blood cells, white blood cells, platelets, and so they're suspended in that plasma. So the second part of the slide, the formed elements, red blood cells, also known as the erythrocytes. We are going to come back much later and talk about the origin of your red blood cells, white blood cells, and platelets, but essentially that comes from stem cells. You do have stem cells in your bone as of now, so as an adult, you have stem cells there that are still able to become those different types of formed elements. White blood cells, also known as leukocytes, and then lastly, I know I mentioned platelets, their formal name is thrombocyte. All right, so I lied. Plasma is 91%. Oh, wait, no, I didn't lie. Plasma is just over 50% of the blood. I hope I'm telling you the truth there. We'll look at a slide in a minute where we fraction out the blood into the different components. But essentially, the plasma is mostly water. But knowing that the plasma is so important to carrying nutrients or organic molecules, as well as salt and ions, that's really what's going to help us with those homeostatic features that the blood is going to give the cardiovascular system. Later on the slide here, I get my little cursor out, plasma proteins. Those are the most abundant organic molecules in the blood. And as I mentioned, some of those proteins will be involved in blood clotting, but there are other, certainly plasma proteins. One of those is albumin, which is a really huge one that we think about when we're treating people, for example, burn victims. We're going to be thinking about making sure we replace their plasma. The plasma proteins produced by the liver, and those plasma proteins are also going to create osmotic pressure in the blood. So that's going to help essentially determine where water is flowing. Is the water moving out of the cells into the interstitial fluid? Or there's also something called third spacing. Where is the water moving? So osmotic pressure is something that's super important when it's being maintained naturally, but it's super important that in a medical field that we think about when we're treating patients who have been in a variety of severe accidents. So also organic molecules, glucose, of course, our number one energy source, feeding our mitochondria so that cellular respiration can occur and we can crank out ATP. Of course, we're sending amino acids around so that we can build up the proteins that we need in our body. And then we know that a liquid waste that we form from metabolism is called urea. And so that's going to be streaming in the blood. Eventually, we're going to get rid of that through the kidneys. All right, so here we go. Plasma is 55% of your blood. And then you can see if we take your blood and we put it in a centrifuge, then we can essentially get results like this. So you see that when we fractionize the blood, 55% is the plasma. So we talked about that. These things that end in INS, OGEN, these are plasma proteins. So they're about 7% of the plasma, but most of it's water. We just talked about this, and then we have some other solutes. When we take a look at this portion here, these are the white blood cells, but most of your formed elements are going to be made up of your red blood cells and platelets, so about 45%. On your study guide, there is essentially some terms that you should know, and essentially it's the proteins that are involved in that cascade of events that occurs when you cut yourself to when you form a clot or a scab. So essentially, as I mentioned before, when you injure your skin, so let's say you're cutting up an apple, or maybe you're out doing some weeding and you got into some what we call briars, or in like rose bushes that have thorns, and you've certainly scratched yourself. Well, that injured tissue is going to, when cells are damaged, actually, they sound this chemical alarm. And that chemical alarm is going to bring clotting factors to the area. So the clotting factor that really starts the cascade of events is prothrombin activator, and it's going to convert, as its name sort of implies, prothrombin into just thrombin. So that's the first thing. Prothrombin activator is going to convert prothrombin to thrombin. Then thrombin is going to act as an enzyme. Now, enzymes are actually proteins, and that thrombin is going to convert the plasma protein fibrinogen to fibrin. And that fibrin, just as its name implies, it's like fibers that is going to form a network for the clot. So you're bleeding, and this fibrin is beginning to form the network that traps the red blood cells and platelets so that you have that scab. So again, a picture is worth 1,000 words, of course. So here we see the steps. You punctured yourself. Maybe you, like I said, pierced yourself with a knife when you're cutting up an apple. The platelets are going to congregate. So that means the platelets are rushing there, and like as an immediate stopgap measure, oh, let's form a plug. But there's these chemical alarms that are going off because you damaged your tissue. So that's when the prothrombin activator is going to convert to prothrombin, and eventually then it's going to initiate some other what they call enzymatic reactions. But essentially what you get is fibrinogen being converted to fibrin, and now we're patching up that particular wound. All right, so a little bit more about our red blood cells and white blood cells. So red blood cells have this really cute little shape. So if you can imagine a donut, but where the hole would be in the donut, it's just like a thin layer of dough. So essentially, when we take a red blood cell, it is both concave on both sides of the red blood cell. What it has done, it has lost its nucleus. And the reason why it's lost its nucleus is it's got to essentially maintain the shape that's optimal for a pigment called hemoglobin. Now, you might not have known that hemoglobin is a pigment. We need iron, or the heme group, to make up that hemoglobin. And that hemoglobin is specialized to carry oxygen to our cells. These are the most abundant of our cells, are the red blood cells. Why? Because we need all that oxygen on demand for our body to do cellular respiration. Remember how I said in the last chapter, chapter 5, that once you get to the capillaries, there's a single file line of red blood cells. And that's so that each one of them has the opportunity to offload its oxygen. And that is the most optimal way to ensure that we are using the oxygen that we obtain from the respiratory system and getting it to the cells as we should. Okay, the white blood cells, which are a smaller fraction, a very much small fraction of the blood, as long as you're not sick, these are going to be our defense against disease. Now, as I alluded to, there are a couple, 2 or 3 white blood cells that are always circulating in your blood. They're not that abundant, but they're there just in case. Macrophages, neutrophils, natural killer cells. They all have a slightly different attack mechanism, if you will. Now, our white blood cells are going to range in shape, And the nucleus is going to give us a clue as to what white blood cell it is. The nucleus is kind of large and it can be lobed, which is a little bit unusual. Generally, white blood cells are translucent, so we stain them. When you look at the picture of them in just a bit, you're going to see that they look light lavender as far as the cytoplasm, and then they're dark purple for the nucleus. But we wouldn't normally see these leukocytes because they're transparent. So we have to stain them. So the number of white blood cells that you have in your blood at any given point in time is giving us an idea of whether you have infection or not. And when we get sick, there are certain white blood cells that are increasing, and so that helps with essentially determining what kind of infection you have or disease that you have, and even like how long have you been sick. So here is a table from this chapter, chapter 6, that shows us some white blood cells. I do not want you to memorize these. You can certainly take a look at how the different ones work. And it turns out that, for example, let's look at this one here, the eosinophil. If you have a whole lot of eosinophils, you could actually be fighting a pathogen like a parasitic worm. Not fun to think about, but certainly that's one thing that they're looking at if you're having severe digestive issues. So take a look at the white blood cells. They have been stained. These here have more of a bluish looking cytoplasm, but when I've looked at them in lab when they're stained, they're often a light lavender. And then we see this nucleus, so this one looks comma shaped, this one just looks like a period, this one almost looks like lungs, this one's similar, this one has 3 lobes. So that is certainly one way to determine which white blood cell you're looking at. Another topic in this part, or in this chapter, is essentially diving down into the details of the ABO blood system, or blood typing system. So here's the story. See, here we have our cute little red blood cells again, and our red blood cells may or may not have any antigens on them. So an antigen is a foreign body, if you will. That's generally how we think about it. It's essentially a protein that's going to cause a reaction. So if you can put that aside for a moment and just think about what you know about blood types. There's type A, type B, type AB, type O. There are other blood typing systems besides the ABO blood type system. So here we're showing a person with type A blood. I have type A blood. So on my red blood cells, I carry a type A antigen with a specific carbohydrate attached to it. So we call this antigen a glycoprotein, a sugar protein. Now, in my body, I'm also circulating in my plasma what we call anti-B antibodies. So what this means is I cannot receive type B blood. It is a defense mechanism. I have type A blood. If I get another type A blood, I'm going to be hunky-dory because my red blood cells have these A antigens. But in my body, I carry anti-B antigens. So if you give me B, then clumping's going to occur, and that could kill me. All right. So a type B person has a slightly different type of antigen. So here they make them look like spikes. Type B antigen has a different glycoprotein, so a slightly different sugar attached to the protein, and we call this type B. Now, if I'm a type B person, then I'm going to have anti-A antibodies. Antibodies attack antigens. So those anti-A antibodies are going to ensure, as a defense mechanism, I'm not receiving any A blood. Now, let's take a look at the other 2 blood types, much rarer. So we have type AB. If I'm type AB, guess what? I have both those round glycoproteins and the spiky glycoproteins. And notice I don't have any antibodies. No anti-A, no anti-B, because if I did, it would attack my own red blood cells, right? Okay, so then we take a look at a person with type O. Type O essentially has no antigens on that red blood cell. So guess what? I can receive type O blood from someone because there's no antigens there. And my body, even though I have anti-B, guess what? It's not going to do anything to a red blood cell that doesn't have, any B antigens on them. Also, if I have type O, I'm going to have both A and B antibodies. So I can't receive AB blood. Let's pause here, and then we'll have part 2 of chapter 6.